The VTM Podcast, Episode 16. De-extinction and gene resurrection. Hello and welcome. There is a strange new sentence entering the language of science. Extinction may no longer mean what it used to mean. For most of human history, extinction was final. A species vanished. A voice left the forest. A shadow disappeared from the plain. A wing no longer crossed the sky. A herd no longer shook the ground. The bones remained. The stories remained. Sometimes the drawings remained. But the living creature was gone. The world had closed a door, and no human hand could open it again. But now, in 2026, that door is being tested. Not opened fully, not safely, not without danger, but tested. Ancient DNA is being recovered from bones, teeth, feathers, hair, ice, caves, sediments, museum specimens, and forgotten fragments of the dead. Genomes of extinct animals are being reconstructed. Living relatives are being compared to vanished ancestors. Gene editing tools are becoming sharper. Synthetic biology is becoming more ambitious. And a new scientific idea has moved from the edge of imagination into serious debate. De-extinction. Gene resurrection. The attempt to recover lost traits, revive vanished biology, help endangered species, and perhaps one day bring back something that looks, behaves, or functions like a creature the Earth has already lost. This is not Jurassic Park. Not really. There are no perfect dinosaurs waiting inside amber. No simple button that brings back the mammoth. No magical cloning chamber that reverses death. The truth is more complicated and more interesting, because the real question is not simply, can we bring extinct species back? The real question is, what does it mean to bring something back? Is a mammoth, elephant hybrid, a mammoth? Is a genetically edited wolf with ancient traits a dire wolf? Is a resurrected gene the same as a resurrected species? Is restoring a lost function the same as restoring a lost life? And if science gives us the power to reach into extinction, what kind of responsibility comes with touching the dead? This is VTM Podcast. I'm Ralph Clayton. And today, in episode 16, we are talking about de-extinction and gene resurrection, the science of lost life, the dream of biological return, and the uneasy frontier where ancient DNA, synthetic biology, conservation, and human ambition meet. To understand de-extinction, we first have to understand what has changed. Extinction itself has not changed. When the last member of a species dies, that lineage is gone from the living world. The animal no longer breeds. The population no longer adapts. The ecosystem no longer hears its call or feels its movement. The world moves on without it. What has changed is our access to biological memory. DNA is memory written in matter. It is not memory in the human sense. It does not remember pain, migration, hunger, fear, or the shape of a winter sky. But it carries instructions. It carries ancestry. It carries the molecular record of what a body was built to be. For a long time, once a species disappeared, most of that information disappeared with it. Bones could tell us structure. Fossils could tell us size. Teeth could tell us diet. Isotopes could tell us environment. Ancient art and historical records could tell us how humans saw the creature. But the code itself was mostly lost. Now that is changing. Ancient DNA research has become one of the great revolutions in modern biology. Scientists can sequence genetic material from remains that are thousands, tens of thousands, or sometimes hundreds of thousands of years old. They can compare extinct genomes with living relatives. They can identify mutations that shaped cold tolerance, body size, pigmentation, metabolism, immunity, fertility, disease resistance, and behavior-linked biology. They can recover not just the shape of extinct life, but some of its molecular instructions. This is the foundation of gene resurrection. At its simplest, gene resurrection means taking a gene, or a genetic variant, from an extinct or ancient organism, reconstructing it and studying what it did. The goal may not be to recreate the entire creature. It may be to understand a lost enzyme, a lost immune trait, a lost protein, a lost adaptation, a lost biological solution that evolution once discovered. This matters because extinction does not erase only animals. It erases answers. A vanished species may have carried resistance to a disease, or a unique metabolism, or an adaptation to cold, heat, drought toxins, or low oxygen, or a molecular mechanism useful for medicine, agriculture, or conservation. When scientists talk about gene resurrection, they are not always talking about bringing back the mammoth. Sometimes they are talking about bringing back a biological function. That distinction matters, because the public tends to imagine de-extinction as the return of a whole animal. But the science is wider. It includes ancient proteins, ancient enzymes, ancestral genes, lost traits, extinct immune systems, rescued diversity, and genetic information that may help living species survive. In that sense, gene resurrection may become less about reversing extinction and more about using the archive of vanished life to protect the life that remains. But of course, the dream that captures the imagination is bigger. The mammoth, the dodo, the thylacine, the passenger pigeon, the dire wolf. Animals that haunt the human imagination because they are close enough to remember, or powerful enough to mythologize, or strange enough to feel like unfinished business. The woolly mammoth is the symbolic giant of de-extinction. It lived across the cold landscapes of the Ice Age, It was adapted to harsh climates. It had thick hair, fat, small ears, and other traits that helped it survive in frozen worlds. It disappeared thousands of years ago. Not all at once. Not everywhere at the same time. But eventually, the mammoth passed out of the living world. Now, because mammoth remains have been preserved in permafrost, scientists have access to remarkable ancient DNA. And because the mammoth's closest living relatives are elephants, the idea becomes technically imaginable. Not easy, not solved, but imaginable. Compare mammoth DNA with Asian elephant DNA. Identify key mammoth variants. Edit those variants into elephant cells. Try to produce an elephant-like animal with mammoth-like traits. Cold tolerance, hair, fat, small ears. A body better suited to tundra. This is not cloning in the simple sense. You cannot simply take a perfectly preserved mammoth cell and restart it. Ancient DNA is usually fragmented. Cells are dead. Genomes are damaged. The cleanest version of cloning requires living or intact cells. And for extinct animals, those are usually unavailable. So the realistic route is not exact resurrection. It is proxy creation. A living animal engineered to carry selected traits from an extinct species. That word matters, proxy. A substitute, a representative, a biological echo. A mammoth, like elephant, would not be the exact same thing as a Pleistocene mammoth. It would be a new organism, shaped by old information. A creature born from modern science. Modern surrogacy or artificial womb technology. Modern conservation politics and ancient genetic memory. It may resemble the lost species. It may perform some similar ecological functions. It may even behave in ways that surprise us. But it would not have the original population. It would not have the original evolutionary history in full. It would not have the original culture of herd behavior. Unless it somehow learned elephant, social life, and developed its own. It would be both return and invention. That is the paradox of de-extinction. The more honest version of the field does not promise time travel. It promises biological reconstruction. And reconstruction is never the same as resurrection. The dodo tells a different story. The dodo was a flightless bird from Mauritius. It was driven to extinction after humans arrived, through hunting, habitat disruption, and introduced animals. Unlike the mammoth, the dodo has no living body plan close enough to simply remake it by editing a few obvious traits into a near identical animal. Its closest living relatives are pigeons. That makes the technical pathway very different. Bird reproduction is not like mammal reproduction. You cannot handle bird embryos in the same way as mammal embryos. You cannot simply implant a bird embryo into a uterus. For birds, scientists look at germ cells. the cells that give rise to eggs and sperm. The idea is to edit the reproductive lineage of a living bird relative so that it can eventually produce offspring, carrying desired extinct-like traits. This is extraordinarily difficult, but it also shows how de-extinction is not one technology. It is a toolbox. For mammals, you may think in terms of edited cells, embryos, surrogates, and perhaps artificial wombs. For birds, you think in terms of germline editing and reproductive biology. For marsupials, like the thylacine, you face another set of developmental challenges. For amphibians, different again. For plants, sometimes seed banks, cloning, tissue culture, and ancient samples may matter more. Every species asks a different question. Every extinct lineage has a different technical lock, and every living relative provides a different key. This is why de-extinction is not one road. It is a map of hard roads, some barely visible. The thylacine, often called the Tasmanian tiger, is another powerful case. It was not a tiger. It was a marsupial predator. Striped, lean, dog-like in outline, but biologically very different from a dog. The last known thylacine died in captivity in the 20th century. That recent loss gives the thylacine a special emotional force. We have photographs. We have film. We have museum specimens. We have memory close enough to feel like guilt. The thylacine was not lost in deep prehistory. It was lost in the modern age. It was exterminated under human pressure. That changes the moral atmosphere. When people talk about bringing back the thylacine, they are not only talking scientific possibility. They are talking about repair, about apology, about whether humans owe something to the species we erased. But again, the science is hard. The thylacine's closest living relatives are not thylacines. They are other marsupials. To recreate a thylacine, like animal, scientists would have to reconstruct a genome, understand which genetic differences matter, edit living cells, solve reproductive and developmental problems, and produce an animal whose biology may still not fully match the original. And then comes the ecological question. Where would it live? What habitat remains? What prey base exists? What diseases would it face? Would the modern ecosystem accept it? Would humans? De-extinction is never just a laboratory problem. It is an ecological problem, a legal problem, a social problem, a moral problem. A creature cannot be restored only into a genome. It has to be restored into a world. This is where conservation enters the story. One of the strongest arguments for de-extinction technology is not that it will bring back extinct species, but that it may help endangered ones. Many living species are not extinct yet, but they are genetically damaged by small population size. When a population collapses, it loses genetic diversity, inbreeding increases, fertility can decline, disease vulnerability can rise, adaptability shrinks. The species may still be alive, but its future becomes narrow. This is where gene resurrection becomes practical. If scientists can recover genetic diversity from museum specimens or from older remains, they may be able to reintroduce lost variants into living populations. This is not de-extinction in the dramatic sense. It is genetic rescue. And it may prove more important. Imagine an endangered animal today, a bird, a rhino, a cat, a marsupial, a frog, a plant. Its living population carries only a fraction of the diversity it once had. But museums hold specimens collected a century ago. Those specimens contain DNA from populations before the collapse. If scientists can identify useful lost diversity, and if editing or assisted reproduction can safely reintroduce it, then gene resurrection becomes a conservation tool, not a spectacle, not a headline animal, a way to repair the genetic narrowing caused by human damage. This may be the sober future of the field. Less about bringing back the dead, more about keeping the nearly dead alive. Less about building a mammoth for wonder, more about restoring resilience to species we can still save. And maybe that is where the moral center should be. Because the danger of de-extinction is that it can become a distraction. People may think extinction is less tragic if science can reverse it. They may think conservation matters less if extinct species can be brought back later. They may accept destruction today because someone promises resurrection tomorrow. That would be a terrible mistake. A revived proxy is not the same as a living ecosystem. A recreated trait is not the same as an ancient population. A few engineered animals are not the same as a species restored across its range. And no technology can replace the ordinary work of protecting habitat, stopping poaching, reducing pollution, controlling invasive species, limiting climate change, and respecting the living world before it becomes a museum of regrets. Now we have to confront the central philosophical question. What counts as the same species? This is not just academic, it shapes public trust. If a company or research group says it has brought back an extinct animal, what does that mean? A perfect clone, a genome level match, a living relative with edited traits, a hybrid, a proxy, a functional substitute, a symbolic resurrection. Species are not just lists of genes. They are populations. They are reproductive histories. They are behaviors. They are ecological relationships. They are developmental pathways. They are microbiomes. They are learned traditions. They are shaped by environment. They are lineages moving through time. If you edit a few genes in a gray wolf to express traits associated with a dire wolf. Have you brought back the dire wolf? Or have you created a grey wolf with dire, wolf-like features? If you build a cold, adapted elephant with mammoth traits, is that a mammoth? Or a mammoth-like elephant? If you produce a pigeon lineage with dodo-like traits, is that a dodo? Or a new bird carrying selected dodo characteristics? Honest language matters. The public deserves precision, because the word resurrection carries emotional power. It suggests return. It suggests miracle. It suggests death defeated. But science should be careful with miracles. In most cases, what we are talking about is not true resurrection. It is reconstruction. It is editing. It is approximation. It is biological design inspired by extinct genomes. That can still be scientifically valuable. It can still be ethically meaningful. It can still help conservation. But it is not the same as reversing time. The dead do not simply walk back unchanged. There is also the question of animal welfare. This is one of the hardest parts. De-extinction experiments would involve living animals, surrogate mothers, edited embryos, failed pregnancies, developmental abnormalities, small founder populations, captivity, uncertain behavior, uncertain health. A mammoth-like calf, for example, would not be born into a world of mammoths. It might be born from an elephant surrogate, or from an artificial womb, if that technology becomes possible. It might need social learning from elephants. It might have a body adapted to cold, but a mind shaped by isolation, human management, and experimental novelty. What does it owe us? Or rather, what do we owe it? A creature created for conservation is still an individual. It can suffer. It can feel stress. It can fail to thrive. It can be displayed, marketed, misunderstood, or made into a symbol before it is allowed simply to live. This is especially important because de-extinction attracts spectacle. The first mammoth-like calf would be global news. The first dodo-like bird would be global news. The first thylacine-like animal would be global news. Fame can distort science. It can attract funding. But it can also create pressure to overclaim, to rush, to brand, to turn living beings into proof of concept. A responsible de-extinction science must put welfare at the center. Not after the announcement, before the experiment. If the animal cannot live a decent life, then the project has already failed at a moral level, no matter how impressive the genome editing looks. Then there is the ecosystem question. Suppose we can create a proxy species. Where does it go? The mammoth is often linked to the idea of restoring Arctic grasslands. Some argue that large grazing animals once helped maintain cold grassland ecosystems by knocking down trees, compacting snow, cycling nutrients, and influencing vegetation. If Mammoth, like animals, could help restore those functions, maybe they could affect carbon storage or permafrost dynamics. That is the vision. But ecological systems are complex. Introducing or reintroducing large animals is never simple. A proxy animal may not behave like the original. The climate is not the same. The plant communities are not the same. Human land use is not the same. Diseased landscapes are not the same. Predators and competitors may not be the same. And even if a restored function is useful in one region, it may be harmful in another. The history of conservation is full of unintended consequences. Species introduced to solve one problem sometimes create another. That does not mean rewilding is bad. It means ecological humility is necessary. A de-extinct proxy is not just a scientific achievement. It is an intervention, and interventions must be tested carefully. Small scale, long-term, transparent, regulated, monitored, with local communities involved, with indigenous rights respected, with ecological uncertainty acknowledged. A species is not a museum artifact that can be placed anywhere. It is a force in a living network. And networks answer back. The deepest concern may be this: de-extinction could change our emotional relationship with extinction. Extinction should frighten us. It should grieve us. It should feel final enough to demand action. If people begin to believe extinction is reversible, the moral pressure to prevent it may weaken. That is why scientists and communicators must be clear. De-extinction does not make extinction harmless. It does not restore the vanished world. It does not bring back every individual. It does not restore lost generations. It does not recover the original ecosystem. It does not undo human violence. At best, it creates a new possibility from fragments, a possibility that may help, a possibility that may teach, a possibility that may partially restore function. But it is not absolution. The passenger pigeon once moved across North America in flocks, so vast they darkened the sky. Billions of birds, a living river. Humans destroyed them with industrial killing and habitat loss. If we one day create a passenger pigeon like bird, that does not restore the billions. It does not erase the slaughter. It does not give back the forests as they were. It may create a symbol. It may restore some ecological role. It may teach us something. But it cannot fully repay the debt. This is why de-extinction must not become a loophole in conscience. It must become a mirror showing us the cost of carelessness. And perhaps, if used wisely, a tool for repair. There is another side of the story that deserves attention. Gene resurrection may help us understand evolution. When scientists reconstruct ancient genes, they can test how proteins changed over time. They can ask what mutations mattered. They can investigate how organisms adapted to climate, diet, pathogens, oxygen levels, or ecological niches. They can resurrect ancestral proteins in the lab, not as living creatures, but as molecular fossils brought into function again. This is powerful. It allows scientists to move from historical inference to experiment. Instead of merely asking what an ancient gene might have done, they can synthesize it, express it, measure it, compare it, and learn from it. This can illuminate the deep history of life. It can show how proteins become more stable or less stable, how enzymes shift function, how receptors change sensitivity, how immune systems adapt, how extinct animals may have handled cold, disease, toxins, or diet. This is not as dramatic as a mammoth calf, but scientifically, it may be more important. The resurrection of a gene can reveal principles, and principles travel farther than spectacle. A revived ancient protein could inspire medicine. A lost enzyme could teach chemistry. A reconstructed immune variant could reveal disease vulnerability. An extinct adaptation could help living species face climate stress. In this version of the field, the dead are not brought back as animals. They return as knowledge. And knowledge may be the most ethical resurrection we have. But some boundaries should remain. Human de-extinction, for example, is a moral abyss. People sometimes ask whether Neanderthals could be brought back. The technical answer is already extremely difficult. The ethical answer is far clearer. We should not create a human or human, like individual as an experiment in resurrection. A Neanderthal would not be an exhibit, not a research object, not a curiosity, not a proof of concept. It would be a person, or at minimum, a being close enough to personhood that all the deepest ethical protections should apply. There would be no consent from the extinct lineage, no natural community, no rightful social world prepared to receive them, no justification strong enough to overcome the harm and exploitation involved. The same caution should extend to any project where consciousness, suffering, identity, and social deprivation become central. De-extinction is already ethically complex with animals. With extinct humans or human relatives, the answer should be no. Some doors should stay closed. Science is not made weaker by restraint. It is made more civilized. So where does the field stand in 2026? It stands in a fascinating middle place. Ancient DNA has become powerful. Genome editing has become sharper. Synthetic biology has become more capable. Conservation genomics is advancing. Private companies are making bold claims. Public excitement is high. Expert skepticism is also high. That is probably healthy. A serious field needs ambition and criticism. It needs builders and skeptics. It needs people who imagine what is possible and people who ask what is true. The biggest breakthroughs in the next few years may not look like full resurrection. They may look like improved ancient DNA recovery, better genome assembly from degraded samples, more accurate identification of functionally important variants, improved reproductive technologies for endangered species, gene editing for conservation, biobanking, artificial womb research, stem cell tools, organoid models for extinct traits, better ethical frameworks, better language, better standards for what counts as de-extinction. The field may mature by becoming less theatrical. That may sound less exciting, but it would be better science. The future should not be measured only by whether a mammoth, like animal, appears on camera. It should be measured by whether endangered species survive, whether ecosystems are strengthened, whether extinct biology teaches us something real, whether animal welfare is protected, whether claims become more honest, whether the public learns the difference between a miracle and an engineered proxy. That is how de-extinction becomes more than a spectacle. That is how gene resurrection becomes a serious scientific frontier. There is a beautiful and terrible irony in all of this. The same species that caused so many extinctions is now developing tools to reach backward into the genetic ruins. Humans destroyed habitats. Humans hunted species into silence. Humans moved invasive animals across oceans. Humans warmed the climate. Humans narrowed the living world. And now humans ask whether science can repair what human power broke. That question is not wrong. Repair is better than indifference. Restoration is better than despair. But repair without humility becomes another form of domination. The old mistake was thinking nature was ours to consume. The new mistake would be thinking nature is ours to rebuild however we please. The right path is narrower. Use the tools, but do not worship the tools. Recover what knowledge can be recovered. Protect what life remains. Restore where restoration is honest. Resist spectacle when spectacle cheapens life. And remember that the first duty of conservation is not to resurrect the dead, but to defend the living. I think that is the heart of episode 16. De-extinction is not one idea. It is several ideas wearing one name. It is ancient DNA. It is synthetic biology. It is conservation genomics. It is gene editing. It is ecological restoration. It is animal welfare. It is public imagination. It is corporate ambition. It is grief. It is guilt. It is hope. And it is a warning. Because extinction is still real. A species can still vanish. A forest can still fall silent. A reef can still bleach into ruin. A bird can still stop returning. A frog can still disappear from a stream. A predator can still be remembered only by bones, photographs, and the shame of those who came after. No technology should make that feel smaller. If de-extinction teaches us anything, it should teach us that life is harder to restore than to destroy. The genome may be recovered. The trait may be edited. The proxy may be born. But the world that made the species, the relationships that sustained it, the generations that shaped it, the wildness that belonged to it, those things cannot simply be printed back into existence. They must be protected while they still live. So maybe the greatest value of gene resurrection is not that it lets us defeat extinction. Maybe its greatest value is that it forces us to look extinction in the face, to see that every vanished creature was not just an image, not just a specimen, not just a name in a book, but a library of life, a solution to the world, a voice in the order of nature. And when that voice is gone, science may recover an echo, but it cannot recover the whole song. That is why this frontier matters. Not because it promises easy resurrection, but because it makes the boundary between loss and responsibility impossible to ignore. We are learning to read the DNA of the dead. The harder question is whether we will learn to protect the living. This has been VTM Podcast. I'm Ralph Clayton. Until next time, remember this. The future is not saved by bringing back ghosts. It is saved by refusing to create them. And don't forget, when we talk about robots or AI, I have to say this, very important. The question is not whether machines can think. The question is whether mankind will remember what thinking is for. Not every invention is progress. Not every intelligence is wisdom. Not every mercy is salvation. Build the machine. Study the machine. Use the machine. But never kneel to the machine. Because when the age of steel begins to speak with the voice of gods, one truth must remain above every circuit, every throne, and every star. Man shall remain man, or perish as himself.